So we wanted to put the presentation in the context, starting with the slide that summarizes one important contribution of transcriptomic studies in cancer immunology. So what these studies have highlighted is that there are redundant signatures that are shared among tumor with better prognostic connotation, or tumor that will respond to immunotherapy, and the same signature are reflected and maximized in post-treatment lesion in tumor that will subsequently respond to treatment. So there are very similar immune models that are shared by this one we call prognostic and predictive signature. So what does it mean? The tumor that will respond to therapy is the tumor that is pre-inflamed, and a certain degree of pre-inflammation is required for the tumor to respond. And how we define this inflammation? So, well, different studies, especially using gene expression, have defined different classifiers. But if you look at the literature, there are always a certain category of gene that is overrepresented over and over in prognostic and predictive classifier. This model reflected a phenomenon that is characterized by the activation of the expression of specific chemokines that are CXCR3 and CCR5 ligand. These are CXCL9, CXCL10, CXCL5 that are important to attract cytotoxic T cell and NK cell on the tumor bed. The second model is the Th1 signaling. So you need a Th1 oriented immune response to be effective, and this one at the molecular level is regulated by the transcriptor factor STAT1 and IRF1 is an interferon gamma mediated polarization. And the last model is the effector function that is represented by granzyme, perforin, and granulysin. And these are the molecule that will mediate tumor lysis. So we call the immunologic constant of rejection because the same pathway are also observed in other form of immune-mediated tissue destruction, like, for example, allograft rejection. So are not specific of immune-mediated tumor destruction, but this pathway are also observed in other similar phenomenon like allograft rejection, flares of autoimmunity, or clearance to pathology. And this is what came up from transcriptional analysis data. So then, when we generate this hypothesis, this was based on group work from our group and the other, and the most of the study were done before the implementation of PD-1 in clinical practice. So, but the recent study in the context of PD-1 confirmed the hypothesis, the observation. So the interferon gamma related transcript can segregate relatively well tumor before treatment from patients that respond as compared to the one that will not respond. And there is an interferon gamma related signature developed and implemented by Nanoscript called the tumor inflammation signature that has a strong overlap with our ICR gene that it was validated in a cohort of 290 patients affected by multiple cancer. by different kind of cancer. And what happened in the tumor is this expression of this pro-inflammatory gene is paralleled by the counteractivation of immunoregulatory mechanism. So what happened is that the immune system reacts against this response with the activation of immunoregulatory mechanism. So at the end, what happened, the tumors that are inflamed co-express pro and anti-inflammatory genes. And sort of based on this observation, we could speculate the tumor could be segregated in two different or opposite immune phenotypes. One that is called inflamed, or Th1 inflamed, that display the activation of pro-inflammatory genes and the counteractivation of immunosuppressive genes. And this is an important concept because until a few years ago, we thought, okay, the tumor that will not respond to therapy would be the one that have high infiltration of Treg, high infiltration of high expression CTLA4, etc., but not. Actually, the one that are that have higher infiltration of Treg actually would respond better because this infiltration is reactive to an ongoing process, immune-reactive process. The opposite phenotype is called silent and doesn't display any sign of inflammation and doesn't display any sign on immunosuppression, so is completely silent. So the first one is associated by favorable prognosis and response to immunotherapy, and the other is the opposite feature. So having said that, so the critical question is why some patients or some tumor develop one phenotype and some tumor patient the other. Of course, this one is oversimplification. Immunology is not black and white. There are a level of gray, but the opposite are these two phenotypes. So if we look at the phenomena of immune-mediated rejection or the Th1 immunophenotype, we could speculate there are three vectors that can influence their development. One is represented by the environment. There is strong evidence that microbiome and especially gut microbiome influence the responsiveness to immunotherapy and in general the immune homeostasis. The other is the genetic makeup of the host bearing the disease, and the third one is of course the intrinsic tumor genetic program. So we start to explore this third vector that is tumor genetic program, and to do so we mine a large data set that is represented by the TCGA collection. In particular, we focus on breast cancer. That is a tumor that is particularly refractory to immunotherapy. So what we did is we analyzed exon sequencing data, RNA-seq data, and SNPs data to infer the copy number variation, and try to see if we could replicate this observation, if we could identify discrete immune phenotype and then compare them in terms of copy number and somatic mutation to understand how the tumor genetic program might influence the development of this immunophenotype. So what we did was fairly simple. So we start with the gene list of ICR gene is a gene of 20, a list of 20 genes that heavily correlate with each other. So we use consensus cluster analysis to to query the data set to understand what is the best separation a more robust separation we could obtain based on 1,000 permutation, and the results was that this data set can be divided in 4 well-defined and discrete groups that we call ICR4, ICR3, ICR2, and ICR1, in which the level of immune activation progressively decreased from ICR4 to ICR1. So then the ICR4 is the Th1 inflamed subtype. The ICR1 is the immune silent subtype. To just visualize this information, we could estimate the density or the level of different leukocyte population by deconvoluting RNA-seq data. As you could see, the ICR4 have very strong level of infiltration of B cell, T cell, cytotoxic T cell, while the ICR1, the immune silent, has relatively low, almost 0 T cell or 0 B cells, and the few are not polarized toward a T helper 1 immune response, but rather against a T helper 2. So these tumors are indeed the opposite, dramatically different. So what about different intrinsic molecular subtype? We know that breast cancer is very heterogeneous disease. And as not surprisingly, we observed that the ICR4, that again, this one is the T helper 1, the hot tumor, the Th1 inflamed tumor, is enriching basal-like. We know that basal-like tumor, triple negative, tend to have higher level lymphocyte. But what was also interesting that 60% of this phenotype is also represented by the other molecular subtype, luminal A, luminal B, and HER2. So although a small proportion of luminal is inflamed, it contributes quite significantly to this group because they are the most numerous subgroup. So, and this is quite important because luminal in general are not even considered for immunotherapy, but if we only based criteria inclusion of for clinical trial only on intrinsic molecular subtype, we probably miss a proportion of tumor that might be benefit to this treatment. So the comparison of the two groups in terms of survival was quite positive. It is not something that we expected based on data on TIL. Again, ICR4 group have much higher, much longer survival as compared to the immune silent. So then we ask what is the difference among these two groups in terms of copy number, mutational load, and mutation of driver genes. So the difference in copy number, one of the most significant was the amplification of a specific chromosome, region 4q21, that it contains the 6C chemokine, including 6CL9 and 11. So there was a focal amplification of this chromosome segment that contained the genes, the 6CL9, 10, 11, that are genes that encode for chemokines that are important for the attraction of Th1 and cytotoxic T cell, and that also included in our signature. So this was the first point. The second is what about the number of the mutational load? We know that the mutational load is associated with with response to immunotherapy. And there was a trend, indeed significant, among ICR4 among the ICR subtype, in the sense that the ICR4, the inflam, had a higher level of mutation. Although the degree of variation was quite high, there is a strong proportion of tumors that have low level of mutation, but that are inflamed, and or a relatively high level of mutation that are not inflamed. And the mutation was also related to intrinsic molecular subtypes. The basal-like and the HER2 have a much higher level of mutation. And we did the associative analysis within each subtype. The significance was lost, except remain marginal significance for luminal, because there are a proportion of lumina that are relatively very high mutation. This influence this association. So what does it mean? This one, this immunophenotype based on the associative analysis is influenced by both the mutational load and the intrinsic characteristic of the tumor. You can also predict the number of neoepitope, and the results are very similar as the one observed for the mutational load. So then we ask if we can identify specific mutation that are associated with this differential immune response. So we compare these two groups in terms of the presence of a selective mutation by mining the exon sequencing data. So what this heat map show? Okay, if you have this, each row is a patient. Is column is a genes. If it's brown, means that this gene is mutated. Blue is not mutated. Okay, ICR4 have much higher level of mutation, as has been shown in the previous slide. What was interesting is were two genes, MAP3K1 and MAP2K4. They were almost exclusively mutated in the poor immunophenotype. In fact, there was this gene had a presence of 20% almost in the ICR1 and almost absent in the ICR4. So MAP3K1 and MAP2K4 we know that are mutated in luminal, and luminal had more anyway less level of immune infiltration and less lymphocyte. But even when this association has been performed within the luminal type, remains statistically significant. So even within the luminal, there was a strong association between MAP3K1, MAP2K4 mutation and immune subtype. So this is a busy slide. I don't want to go into detail, but we try to understand what is what was the level of activation in this tumor, and what the analysis suggested that the mutated tumor had a relatively activation of overexpression of the final mediator of the RAS MAP kinase pathway. This is an overview of the different MAPK pathways, and these pathways has been shown to downregulate MHC and also induce the production of anti-inflammatory cytokines. Again, these two genes are represented here, and the effect is impossible to assess the effect of the mutation on a specific pathway, because they are extremely overlapping and interconnected. But overall, what the analysis suggested that tumor with this mutation had a strong dysregulation of the MAP kinase pathway. So what is the next step we did is try to calculate a sort of MAP kinase dysregulation, MAP K dysregulation score. And without going to detail, this score could segregate quite well the luminal and the other subtypes in terms of immunophenotype. So that as higher is the MAP K dysregulation score, as low is the intratumoral immune response. So what is the potential implication? That we know that the cold tumor are not going to respond in general to immunotherapy, and one can speculate that the modulation of this MAP kinase through MEK inhibition could convert the immune silent phenotype to the inflamed phenotype, and that can in turn be sensitive to immunotherapy. And this is the end.